Okay, for the final presentation of today, we have biotoxicity for the third time in a row. So biotoxicity is basically anything that's poisonous to cells and bacteria. In our case, we use silver and testing against yeast. So
in the picture here, the yeast is represented by our terrarium greenhouse thing, and the silver particles are represented by the bees, the hive, and the truck of bees.
So why silver is so reactive to so bad for cells is because once silver interacts with the cell, it creates reactive oxygen species and causes lipid peroxidation, which destroys the cell membrane and leaves you with a very dead cell. Now, in our experiment, we messed with 3 different sizes of silver, and each had different levels of toxicity towards the cell. Yeah, what would you like to explain?
So, in our picture, our 3 different sizes are the bees individually, which represent the ions, and they can get in through the little door, but they can only go in one at a time, and they're tiny, so they don't do a ton. You've got the hive representing the nanoparticles, which can get in and release a ton of bees, and they can pollinate everything. And you've got the truck, which is too big for the front door.
So, in order to check how well the yeast cells were doing, we would have a manometer which would detect how much CO2 the yeast cells were producing. Would you care to explain?
All right. So, in our analogy here, instead of having the yeast cells being killed and then they produce less CO2 because they're dead, it's the inverse. And so when you have a bunch of bees all pollinating in the greenhouse, they produce lots of honey, and our kid here is happy. With the single bees, they produce less honey because there are less bees, and kids mildly happy, I guess. And the bee truck can't get in, and so no honey was made.
Now, while we do like the picture and how it displays the concept pretty clearly, we think more should have been added. For example, it doesn't really show how the sizes of silver actually change in color due to plasmonics and stuff like that.
All right. I think that this could have been put into the drawing via sound or representation of sound. So the ions in solution are so small that you just can't see them, and so this could be represented by the bees being so small that you can't hear them. And the nanoparticles will change the color of the solution because they absorb certain frequencies of light. And this could have been represented by the hive making a soft buzz, just to show that there was color there. And the bee truck, representing the macro silver, could just have an engine sound, a rumble or what have you, to show that it can be seen.
Is that about it? Any questions?
Yes.
So you mentioned this is the inverse of what the experiment was like.
Yeah.
How would you change the depiction to make it not the inverse?
So the question is, how could we change the depiction to make it not the inverse of how it works in the yeast cells? And I think we'd have to change the entire premise of the drawing. This is just the inverse. So I don't know how you'd do an inverse with bees.
Is there anything that the bees could hurt or destroy as opposed to flowers that they would pollinate?
I mean, you could probably get the bees stinging animals inside the greenhouse. But I'm not sure how you would show the measuring of that. I don't know.
Maybe some insects that eat the flowers or something like that instead.
Yeah. Insects that eat the plants rather than bees definitely could, and then you could, I don't know, represent this with crops, and say locusts or flies or whatever are coming in, and then the yield from the farm would go down as you got more of the insects coming in. I think that would work. Any other questions? Yes?
In the picture on the bottom right corner, there is no bees going in. The experiment, theoretically, would there have been any quote-unquote bees that would have gone in?
Yes, there would have been a few going in, and they would have released a few bees, but it is, as far as the output is concerned, it would be pretty much negligible, and I think that's what this was trying to show.
Any more questions from the audience? Maybe one more audience question?
Yes.
Oh, I guess there's
One of
two
you.
more audience questions, so we'll do your question first.
Yeah, I'm a little bit confused. The ions are smaller than the nanoparticles. How come more nanoparticles are going in than the ions? I
Okay.
understand that part.
So, even though the ions are smaller than the nanoparticles, they get in through different methods. Like, you can see here that the bees are getting through this little window, and the hive is going through the chimney. In the cells, the nanoparticles will get in through an active transport method, since the nanoparticles are both neutrally charged and about the same size as the food the cell eats. It will, since cells aren't very smart, they will take in this nanoparticle as if it was food, and then the nanoparticle can do whatever. But with the ions, they're positively charged, and cells have gotten very good at dealing with keeping out ions that are unwanted using their charges, and that's why not many can get in.
And then as a quick follow-up to that, so once the nanoparticles are actually inside the cell, why are they so good at releasing ions?
Well, since they're nanoparticles, they have a really high surface area, and they can, that allows a lot of the silver to react with the oxygen in the cell and create those radical oxygens that do the damage. And the silver ions individually can't do that because they're only one ion, and it can only react once.
Last question?
Are there any practical applications of silver nanoparticles?
Yes, they can be used, since they kill cells, for disinfectant type things, and they won't be too terribly deadly to humans because we have so many cells in us that you just need tons and tons of silver to do damage, and you wouldn't do that with the disinfectants. And yeah.
I'll just make a quick addition or edit to that statement. So human cells actually don't tend to endocytose or eat nanoparticle cells in the same way that bacterial cells do. So even if we had a lot of silver, it may not damage the same way because our cells likely don't uptake the silver nanoparticles in the same way. So first, Cruz, I'm so sorry. Your last name auto-corrected on my thing, so I'm sorry that your last name was spelled incorrectly. And then secondly, will the artist of this drawing please stand up so we can acknowledge you?
No way, no way.